Well, I'd like to welcome everybody today to our exciting webinar on corn nematodes. My name is Steve Gauk. I'm the Regional Agronomy Manager for the East with Beck. And today I am excited to introduce to you guys Matt Montgomery. He's our Agronomy Education Lead. And also Janelle Myers, who's our Seed Enhancement Lead at Beck. And they prepared a fantastic webinar today with the corn nematodes for all of you. Going to kind of break it down into 4 different sections here to really get in depth with management, learning about nematodes, which I'm excited about. I want to remind you though of a couple things. At the end, there's going to be a QR code that everyone's going to get the opportunity to scan and get CEUs for their CCA points. And we're also going to have some questions at the end. So be thinking about questions, write them down. There's also, you'll see a chat group within this WebEx webinar that you can type in those questions and I'll follow along. Keep track of those at the end for everybody. So we're going to hold the questions till the end. And then I'm also going to ask everybody a favor. If everybody could make sure that their computers are muted, it really helps with keeping the audio clear and things like that for our speakers. Helps you listen better. So if you would, take a look at your computer, make sure that you are muted from your end. And then, like I said, at the end, we can open it up for questions. So with that, I'm excited. And Matt, we're going to turn it over to you.
Okay, I'm going to do a quick sound check, make sure I get a thumbs up on that. Looks good, good. And then I'm going to go ahead and share my screen and we'll get started. Okay, unless I hear otherwise, I'm going to assume that you all can see my screen. And we're going to get started with the subject of corn nematodes. Thank you, Janelle. I appreciate that. Again, great to be with you today. Matt Montgomery, Agronomy Education Lead. Before we get started with the subject of corn nematodes, I think in the advertisements that we had, we talked about the experts we're going to be on here today. And I'm going to say that probably qualifies for Janelle. I'm not sure that qualifies for Matt Montgomery. A really interested individual on the subject is who Matt Montgomery is. And really should, before I begin here, acknowledge quite a few people that over the years are the experts who have really helped me better understand this subject. So I can think way back in the day when I started my extension career, a gentleman by the name of Dale Edwards at University of Illinois that some people may remember, who was really kind to take us young guys under his wing and teach us a lot about this subject. I think about a master's class I took with a gentleman some of you may remember by the name of Dr. H. Walker Kirby, who was plant pathologist at University of Illinois at that time and taught the class on nematology that I took. Think about Terry Niblick, who was U of I, eventually the Ohio State University. Think about Suzanne Bissonette, Dr. Bissonette, who worked out of the Champaign Extension Center. And then more recently, just some really kind with their time moments with Dr. John Schroeder at University of Illinois and Nate Schroeder, I should say, at University of Illinois, and John Eisenbeck at Virginia Tech. And so just really grateful for the way those people have guided and tweaked and helped over time to put in everything that's part of this presentation right here. You know, as I'm thinking through this today, I think it's important for us to have an outline to go through. And these are the things I hope that I get covered by the time that we're done today. And we're going to start out with this idea of why in the world should you even care about this subject? I'm guessing 85, 90 on the call today have heard the term nematode. But it may be no deeper than that, and that's very understandable. And people really haven't shown you a nematode in the past. Nobody's ever really pointed at an issue in the field and said nematode or corn nematode. And so you're probably justifiably asking, why in the world should I care about this subject? And so I want to jump into that to begin with. I do want to talk about what extreme symptomology can look like. So at the extreme, what are the things we might notice in the field. I want to talk about the appropriate way to monitor, and Janelle's going to jump into that a little bit more during her half of the presentation. Then I'm going to take a pause. I'm going to step back and I'm going to talk about what is a nematode, because we're going to get that deep into the presentation and really need to define what a nematode is. Then I want to talk about plant parasitic nematode anatomy. And then I'm going to pause there for a second, and I'm going to do something that my dad did when he used to teach. Dad used to teach, and at the end of every day, he'd have on the board the must knows, the things that he thought people needed to remember out of that lecture, the most important takeaways. We're going to then go from that into core nematode species. We're going to list those out, and we're going to go through the must knows on that as well. So let's jump in, and I'm going to go ahead and make sure I do this by arrow here. There we go. Why should you care about this subject? Again, 85, 90% of you, I'm guessing, have been exposed to the topic of nematodes, maybe just at the word, really haven't jumped into it any deeper than that. And you justifiably wonder why you should care. And let me pause, take a moment here, step into a different project that we're working on at Beck's. And I'm going to actually reference root reveal. I'm not going to jump in deep to the subject of root reveal. There are videos online. Individuals are going to be talking about that more here in the future. You kind of have an idea if you've been paying attention to Beck's for a while that what we're doing in this project with Root Reveal is we're actually taking hybrids, brands that we sell, and then growing those out and getting an idea of root morphology and figuring out how we pair that up with management. What management tweaks can we look at compared to that root system that help push our input dollars to more efficiency, help us get more bang for buck out of the dollars that we use. I think any of us that have been exposed to root reveal, in the first 5 minutes, we look at a picture like what you see on the left-hand side of your screen there, and we pause and we go, wow, I just don't think I understood how extensive the root system of a corn plant was to really perform at its optimum. I keep trying to fix pictures like this in my mind. For all the other things we've talked about with root reveal, I keep thinking about this is the kind of root development that we have to have, this picture of a root system on a healthy corn plant to make sure we can grab a hold of nutrients and scavenge moisture to make it through a growing season. And I begin to think about what things am I doing that are pulling that root system back from this optimum, that's keeping the plant from having that optimum root system, and undoubtedly is beginning to trim back yield potential. And so I think about what are we doing with tillage? Maybe I'm appreciating just a little bit more how insects, actually their feeding can damage that root system and pull us back from the crop operating in a way that helps us maximize dollars coming into the farm. Begin to think about diseases just a little bit more. And because corn nematodes are root feeding pests, it makes me think about them as well. And that's an important takeaway. We're going to talk about those must knows later on, but I'm going to pause every once in a while and just reiterate those points. I want you to walk away from this and remember that plant parasitic nematodes on row crops are root feeders in the Beck's footprint. I think also one other thing that's really coming home for us is that it also matters what that root system looks like early on. Our root reveal team actually did this. They took a picture of a second stage, third stage corn seedling, and they did the same process where they wrap up that plant in this mesh, or in a column in this case, with the medium that we grow those plants in, wash away all that material. And this is the root system on a V2, V3 stage corn plant. And again, I think we all dig up a root system. We don't realize how much of the roots that we prune off during that process, but just pause and look at that screen and look at how extensive that root system is. This is the kind of early start that a healthy corn plant needs. And look how deep those roots delve. And again, I just want to remind us that I think this project is helping us to appreciate not just that full in the middle of the season, deep into the season, root quality and quantity matter for yield, but it actually begins to influence the path that we're going to take early on. And if we compromise that quality and quantity early on, we're going to have a full season impact on yield that we'll see with the combine. And again, with corn nematodes, with plant parasitic nematodes being root feeders, that's why this subject matters. Row crop plant parasitic nematodes are root feeders, and that means that they are potential yield reducers. They can pull back the quality and quantity of the root system, and that means they're worth paying attention to. So that's the first takeaway here. I want you to remember that they're root feeders. They're feeding on the root system, sucking juices out of that root and compromising yield potential. So let's talk about some extreme symptomology when we're visiting about corn nematodes. Again, we'll get into the subject of what corn nematodes, plant parasitic nematodes are here in a second, but let's talk about symptomology at the extreme. In the most extreme cases, you may see something like this, and I could hop in my vehicle and drive 30 minutes to this exact location. I looked at it a few years ago, and what you're going to notice are kind of stunted, irregular stunting of plants in patches that may be a couple dozen feet wide, or maybe even a little bit larger than that, actually taking up sizable portions of the field. But the plants are going to kind of be irregular sizes. This is extreme above ground symptoms. So in this case, when we ran into this situation, we started working through the list and looking at different things that could possibly explain the stunting that we were seeing, and we began to ask questions. We talked about the herbicide program. It really didn't fit for what we were seeing here symptom wise. We talked about soil fertility, and honestly, this kind of symptomology doesn't fit in this part of the world with some kind of soil fertility issue, and their numbers were pretty good anyway. We couldn't really find any good examples of insects or diseases. We couldn't find another explanation, couldn't find any evidence of compaction or anything that might be at play. And that kind of whittled things down to the point that we went, I wonder if corn nematodes might be at play here. And in the pictures that we showed just a second ago, I think you appreciate that we only see one half of that plant when we're looking at the above ground portion of the plant, right? When we're looking at the plant above ground, we're only seeing one half of the story. And so you always dig a plant up. And that's what we did in this case. And sometimes in these extreme cases, and this is what we saw in this case, these are the symptoms that you might run into. Now, think about that picture of a V2, V3 stage corn plant and how extensive, how surprisingly extensive that root system is on that second, third leaf stage corn plant. And then take a picture on the left and notice how nematodes, those root feeders, have nubbed off those roots. We don't have those root hairs developing like what you saw in that picture of kind of an optimum root system. You can see right there, there's some kind of dramatic difference that's going to have a dramatic impact on yield. I always think the picture kind of on the right there is interesting as well. In sandy soils that are bone dry, one of the symptoms that you may run into, again, if we see extreme levels of symptomology, is that you may have bone dry sand. I mean, not a stitch of water in it at all. And then you dig up that plant and it has mud balls all over the root system. And that is because the corn nematode is so fed up on those roots that they're leaking out juices and forming mud around that root system in an otherwise dry soil. So looking for these little clumps of mud in a dry soil sometimes is a symptom we might run into. But again, it's a symptom at the extreme. This is an extraordinary population really hitting that crop hard. This is kind of at the extreme level. Sometimes you'll see something like this, and I pulled this out of my master's degree class material. Again, you look at this root system and think about what we saw with root hairs, and you'll notice you have clubbiness to the root system. We don't really have root hairs. They almost appear to be clubbing off, almost as if, and they were fed on the root tips, and then they kind of swole up in those regions and produced a bunch of growth where that nematode was feeding. Again, this is symptomology at the extreme, so you do need to know about it. in case you might run into this in the field, you might be able to spot these kind of symptoms and begin to ask questions about whether things like corn nematodes are at work. But with those root feeding pests that we're talking about in this case, it's really important for us to remember that really good monitoring really requires soil sampling. Sometimes we don't have those above ground symptoms, and yet we still have impressive levels of nematode feeding on corn plants. Sometimes we miss those below ground symptoms, and yet still are suffering some yield losses from this pest. And so maybe one of the things to take away from today is not just that these are root feeding pests that suck juices out of the root system, compromise roots, and therefore compromise yield potential. Also remember that the really good way to monitor these things is through soil sampling. So you're going to have to send that soil sample in. Janelle's going to talk about that a little bit more and actually have it analyzed to see what we're doing for populations. So we jumped in here just a little bit talking about nematodes, plant parasitic nematodes being root feeders in the Beck's footprint. We talked about the fact that they're attaching to the root and sucking juices out, sometimes to the point that they're wounding the root system such that it's leaking out material. And we talked about some of those extreme symptoms. We talked about soil sampling and the importance of that. But we really should stop here for just a second and really get the subject of nematodes defined. What is a nematode? Well, let's start out with the idea that nematodes are the most numerous multicellular organisms on the planet. Now, sometimes we'll say that about insects, but insect populations are dwarfed by the population of nematodes that exist in the world around us. And there are all sorts of different types of nematodes. Some of you may be familiar with animal parasitic nematodes, so things like roundworms, or if you've heard about things like river blindness in third world countries, that's a nematode caused disease. If you've ever seen pictures, I know this is a little gross, but again, in a third world country where somebody has a swollen limb because they're infected by a worm, so to speak, that's oftentimes a nematode that's causing that issue. We have certain nematodes that are saprophytic. They're crawling around feeding on dead stuff, and we're thankful for them because they're helping out with decomposition. Then we have some nematodes that are actually predatory. They will hunt down and feed on other nematodes and consume them for lunch. And then we have plant parasitic nematodes, and that is the subject again that we're going to cover today. So one of the things you'll notice in the diagrams that I've drawn here are that nematodes look worm-like. And I should pause for just a second. I've introduced false color into these drawings, so it makes it easier for us to spot structures. If you looked at this under a microscope, you'd just see the lines, the colors wouldn't be there. They'd all be washed out. It would be gray, and you'd still be able to see those structures. I just introduced color to make it a little easier for our eye to spot those. But they are eel-like or worm-like in appearance, but they're a little different than things like earthworms. If you were to look at the cross-section of an earthworm, they have organs in their body, and those organs are all stitched together with tissues, right? There's connecting tissues between the outside, that kind of epidermal material, that cuticle of the earthworm, and then it stitches that tissue to the internal organs as well. If you look at a nematode under the microscope, it's not like that. Nematodes are basically microscopic bags of water with organs that float around within. So they are worm-like in appearance or eel-like in appearance, but they don't have those connective tissues. They're tubes of water, and that means if their body gets snagged, for instance, they'll bleed out that moisture and they will die if they lose that integrity of that cuticle. If there's a snag or a cut, they're going to bleed out and die, and they won't be able to recover from that. So some species are going to produce males and females. It's kind of a complicated world when it comes to reproduction and the nematode. Other species are going to be predominantly female, and they're basically going to produce carbon copies of themselves. Some cases, believe it or not, if the population gets too crowded, females will revert over to being males. Sometimes later on, the males will decide to go back to female. It is a really complicated world when it comes to nematode reproduction. But probably the important little nugget to pull out of this today is that plant parasitic nematodes are small, really small, microscopic. If you were to type a message and push that period button at the end of a sentence, that's about the length, plus or minus, of most of the nematode species that we're going to talk about today. So it's really hard to spot those with the unaided eye. You have to have some kind of magnification to spot them. You're not going to be able to point at something in many cases. There's an exception or two that we'll talk about at a different date, but in many cases, you're going to have some kind of magnification to be able to spot them. You're not going to probably be able to spot those with the unaided eye. So again, let's pause for a second. We're talking about a root feeding pest, sucks juices out. We're talking about sometimes missing above ground symptoms, needing to soil sample to actually catch plant parasitic nematodes. They are microscopic tubes of water with organs floating within. You're going to have magnification to spot them. So with that, let's jump into the subject of plant parasitic nematode anatomy. We are going to go through this. Not every single thing that you see on the screen are we going to talk about. We're going to try to keep it somewhat simple, but I want to get some of the major structures out of the way. So we're going to start with, for today, what I've called the upper digestive region, and you can see something called a stylet there. There are what we call lips. Those are used to suction the nematode onto the plant so that it can kind of anchor itself for feeding. There's something called stylet knobs. We're not going to talk about this DEGO thing. There's a median bulb. We'll talk about some of these, not all of these. And one of the things to keep in mind with a plant parasitic nematode is, maybe if you had to think about something you're a little bit more familiar with, it's kind of like a mosquito. You know, mosquito has a needle-like mouth part that it jabs into you and sucks blood away. That's kind of what the nematode, plant parasitic nematodes, do when they're feeding on the root system. They have a needle-like mouth part, in most cases a hollow needle-like mouth part called a stylet, that they will jab into the root and puncture cells, suck juice out of. They use that stylet, that needle-like mouth part, to suck those juices that are going to be needed to sustain them. So that stylet, sometimes actually under the microscope, you can see the stylet with some species rapidly moving in and out of the nematode's mouth, jabbing that root, damaging it so it can slurp up the contents. And that means, to be able to do that, it's got to have some muscles that are connected to it. And so sometimes you'll notice two little knobs at the bottom, and there are muscles that are connected to that that help that stylet move in and out of that plant root jabbing cells. It's also kind of important to keep in mind that nematodes are kind of like flies in the way that they feed. So if you're familiar with how a fly feeds, it basically regurgitates digestive juices outside of its body, and then that partially digested material, it kind of sponges up. The nematode kind of does something similar to that. So as it's feeding on that root, it's going to squirt out digestive juices into the root system. It's going to partially begin to decompose, to break apart, I should say, that material. And it's going to take that nutrient-rich slurry and slurp it back up through the stylet to sustain itself. So a little bit like a fly, it's squirting digestive juices into the root, partially digesting that material, sucking it back in through that needle-like mouth part. And then we should probably go on here now to talk about what I'm going to call for today the lower digestive region. And we are not going to talk about every single structure here. I'm going to flash them on the screen, but let's talk about the important stuff. Now, I mentioned that it's sucking things out. And there's a structure that actually, moved just a little too fast. I apologize for that. I need to talk about this picture right back here. This is what I need to discuss. This is called a median bulb. And the best way I can describe the median bulb is that if you've ever had a little kid, and you know, they send you home from the hospital after they're newly born, they might give you that little blue squeegee thing that you use to get crud out of their nose, right? That's kind of what a median bulb is in a nematode. It's this little structure, and the size of that internal region is manipulated. It's kind of increased or decreased to either force stuff out or to create that suction to pull things back in. And I mentioned that, and I've gone back to show that because in some pictures you're going to see here in a bit, we're going to see some of these structures on certain nematode species, and you're going to see it's not just a drawing. This is kind of real world stuff. Let's go back to that lower digestive region, and I'll get back on track here. Those digestive juices are created in this little structure called a basal bulb. Those digestive juices are then shot back up through that needle-like mouth part. And of course, at the end of the day, that nutrient-rich material that is pulled back in through that needle-like mouth part is passed down into an intestine, and all the good stuff is absorbed out into the nematode's body, and then waste material is funneled back into the intestine so that it can be shot out through an anal pore so it can get rid of that waste material that it's producing. So before we move on here, a couple other things that are important for us to review, right? So we talked about the way that they feed. They're punching the root with a needle-like mouth part. They're partially digesting that material outside of their bodies and taking that nutrient-rich slurry and sucking it back in. They're doing that with this little bulb-like thing that increases and decreases in size to create some of that suction. And then all that nutrient-rich material is going down to the intestine so that it can sustain the nematode and maintain life. Now, all of that means nothing if the nematode can't move and find the root to begin with. And so we're going to talk about mobility or seeking structures here just a little bit. And we're going to begin with some structures that exist on the front of the nematode, on its head, and sometimes on the tail end. You think about the nematodes crawling around in the pitch black soil profile, right? These plant parasitic nematodes that we're talking about. So it's not going to be able to see anything anyway. It doesn't have any eyes. How is it finding the root? It has little structures on the front of its head, on the lip region there, on the head region, that kind of help this thing act like a heat-seeking missile. So as roots are leaking out chemicals, what we call root exudates, those little sensors pick up on those exudates, and the nematode kind of turns toward it as it feels, senses just a little bit more of those chemicals being released by the root. And then it's going to turn toward it, and that signal is going to get stronger because it's encountering more of those chemicals. And it's going to just keep honing in on the root using those little structures. Sometimes those things that we call amphids that help it find those chemicals being released by the root are accompanied by similar structures on the tail end that are called phasmids. Now, movement is controlled by a nervous system. There is a crude brain in a nematode, what we call a nerve ring, and that's really what it is. It's just a ring of that kind of material. And there's a nerve cord of nerve fibers, I should say, a ring of nerve fibers. And there's a cord that runs down the back of the nematode and down the belly of the nematode, and muscles directly connect to that cord. And then, of course, the muscles need something to pull against. You think about you and I, our muscles don't mean a lot if they aren't pulling against a skeleton. There's no skeleton in the nematode, though. So those muscles need to pull against something a little bit more rigid. And what they pull against is basically a hydroskeleton, kind of this water pressure that makes different parts of the nematode's body a little bit more rigid. And that's what the muscles pull against. Now, I mention all that, and an aha moment should probably be happening for you that are attending the call today. I said that there's a neuromuscular system. And that means that there's probably something that we can use as a nematicide that targets the neuromuscular system, that shuts down their ability to kind of send signals and move the way they need to. And if you're thinking about, well, boy, that sounds like some of the insecticides that we use, that's right. There are some insecticides that are also labeled as nematicides. And what they do is they shut down this ability of the nematode to kind of send signals the way it needs to move to be able to find that root. We're going to jump into reproduction here as we're talking about plant parasitic nematode anatomy. We're going to begin with the male. I'm not going to cover every single structure that's on here, but I am going to mention two things there on the tail end Male nematodes often have these little flaps on the tail end that are called bursa, and they actually grab a hold of the female so that they can mate. And then there are these little forceps-like objects in the tail end that are called spicules, and that helps to make sure sperm gets where it needs to go. The female looks a little bit different. You'll notice she doesn't have the bursa. She doesn't have those spicules. She has an ovary, right? Sometimes two ovaries. And this brings up the subject of why this kind of is important to a nematologist. Why does a nematologist need to know about all these structures? Nematologists use this for taxonomy. They will look at the length of that basal bulb there up front. They'll see whether it has that pumping bulb that we talked about, that orange colored thing up there toward the front. They'll look at how many ovaries that there have. They'll see how things are overlapping on the body, and they'll use this for species identification. And actually, it's really important to get kind of that genera, that genus, that species down for a nematode because it influences the kind of management options that are available for us when it comes to the plant parasitic nematodes to corn nematodes. The female produces eggs. She can store up sperm for a long time. I think in some cases a few weeks. So she can kind of use that to spoon feed sperm to those eggs for fertilization. Those eggs will oftentimes kind of be exuded from the female in a gelatinous mass with other eggs. And this is the life cycle that a nematode goes through. So it has an egg stage. It looks under the microscope like a little gel capsule. And in that gel capsule, it goes through the first of its juvenile stages. And so if you look under the microscope, you can see that little juvenile eel-like thing curled up inside of there. So stage one is a juvenile inside the egg. Then we have stage two. It hatches out of that egg as a second stage juvenile. And if we're talking about some of those that burrow into the root and stay there, a lot of times that's second stage juveniles that are doing that. So it's the second stage that will bore into the root oftentimes and cause that injury. They have 4 juvenile stages. And the way you know they have 4 is every time they pass into the next juvenile stage, they shed their skin, they molt. And so that lets them move on to a larger size, kind of like what insects do. And then you hit the adult stage and it all rolls over again. Okay, so a couple things to keep in mind here. We talked about these are microscopic. They feed through a needle-like mouth part. They're feeding like a fly, digesting stuff outside the body, slurping that material back in. We said sometimes they're females, sometimes males, sometimes you can just have females. We said the female can store sperm for a while. She'll squirt all that material out in a gelatinous mass, and then it goes through this egg to juvenile to adult growth stage life cycle. Let's talk about potential nematicide targets. Some of those should become a little bit more clear because of the subjects we've talked about. Janelle's going to talk about management a little bit more. What we talked about earlier, uh, there are neuromuscular targets. There are things that target the nervous system and shut it down and decrease the ability, kind of inhibit the ability of that nematode to do what it needs to do to stay alive. There used to be a whole bunch of those in the marketplace. So you may remember products like Furidan, Timic, that have kind of rolled off the marketplace over time. Counter would be an example of a product that's labeled as a nematicide. There are another product or two that are actual neuromuscular targets that are influencing the nervous system that we could mention as well. But that's one potential target. Another potential target is that sometimes we apply things that actually create a physical impediment to mobility. So they might form some kind of shell of goopy material that makes it hard for the nematode to actually get to the root. Sometimes, at least listed theoretically, are things that are called molting inhibitors. So they keep the nematode from shedding its skin, and then it can't grow the way it needs to progress through its life cycle. Again, as I've talked to some other people like Dr. Schroeder at U of I, he's not really sure that there are any of these kind of nematicides labeled at this point. Some nematicides target cellular respiration. And a good example of that are SDHIs. And that sounds really familiar because SDHIs are actually a fungicide family. If you're used to three mode of action foliar fungicide products, a lot of times those have an SDHI in them. That's the third component. And the way SDHIs work is at the cellular level, they shut down the ability of the cell to harvest the energy it needs to keep the cell alive. And not only does that work really well at killing off fungi, but it also works really well at killing off nematodes. And we know there are at least some cases in this SDHI fungicide family where some of those products work very well as nematicides. And then we have what we might call multi-target or nondescript nematicides. Sometimes we just don't have a clue how they work. We just know they work. Sometimes they're messing up so many things in that nematode, that plant parasitic nematode's body, that we just can't keep track of them all. So that's our walkthrough on what nematodes are, plant parasitic nematode anatomy, the first bits of discussion on management. I want to go through the must-knows on this section. So out of everything that I talked about, these are the 7 points that I want you to kind of fix in your mind and take away from today. Plant parasitic nematodes are worm-like tubes of water, and they're microscopic in size. They feed and damage with a needle-like mouthpart, and you can imagine why that's bad for the plant. They are root feeders in row crops in the Beck's footprint. They partially digest the material that they're going to consume outside of the body and slurp it back into the body. We didn't talk about this, but when it comes to corn nematodes, many, not all, but many of these are native. They grew up on grass prairie, and then we just introduced another grass called corn, and so they feed on corn while they're at it. And so they're actually more distributed than I think what many of us have appreciated in the past because they were here from the beginning. From the moment we had those grass prairies, they were feeding on those grasses, those grass root systems. Then we introduced corn, and they've just hopped over to the crop. They are best detected with soil sampling. And then remember, we have different nematicides that are available. We have neuromuscular nematicides, SDHI nematicides, some that physically inhibit the ability of plant parasitic nematodes to reach the root, and we have multi-site nematicides as well. So that's our walkthrough on plant parasitic nematodes. Let's take a second or two now, a few moments, to talk about corn nematode species. The first thing I want you to keep in mind is that corn nematode is an umbrella term. Some of you are familiar with the term soybean cyst nematode, right? A soybean feeding nematode that feeds on the root system. And in a future session that Janelle and I are going to work on, we'll have a soybean cyst nematode webinar at some point in the next year or so. We're not going to talk about that today, not in depth at least. But soybean cyst nematode is a specific species. It is one critter that feeds on the soybean plant. Corn nematodes aren't like that. I think at the count and some of the material that I was able to look at, there's better than 4 dozen different species in the United States of plant parasitic nematode that feed on corn. And to kind of keep it simple, we use this umbrella term called corn nematode to just kind of lump them all together. But they are different, and there are a bunch of different genera, there are a bunch of different species, and that means that management is not necessarily as clear cut all the time as it is with something like soybean cyst nematode. There are some very specific things because we're dealing with one thing that we can recommend for soybean cyst nematode that don't necessarily work for corn nematodes as a whole because we've got a bunch of different critters that fit under that umbrella. So I'm going to go through and list these, the big groups. I'm going to kind of talk about what the asterisks are and what that little dotted line down there at the bottom is. And I'm going to do this from largest to smallest. So again, it's not that I want you to memorize these. I just want you to hear the names and tuck them away because maybe someday you'll hear these names and you'll remember, that's a potential corn nematode species. So we have needle nematode, sting, dagger, lance, spiral, in some places root knot, stubby root, stunt, lesion, ring, and pin. And I put the asterisk there because in some ways we have to have a way to tame this conversation down. And so I'm putting those down as ones that can be, but aren't really ones that we run into as often in the Beck's footprint. And then I put a dashed line down there above pin because frankly, I'm just not going to talk about pin at all today. I needed to keep it simple, or as simple as I could. Now, to give you an idea of size, I'm going to reference everything we talk about versus soybean cyst nematode, because even though we're not talking about that today, soybean cyst nematode is the pest that's probably at least a little bit more familiar to many of you. So a soybean cyst nematode, it is the size of a period at the end of a sentence. And so here's a picture of some of those cysts on the root. You'll see that I have a ballpoint pen, the tip of it there on the left, and you see how small those little cysts are there in the kind of off-center part of the picture. Again, about the size of a pencil point. And we're going to reference this size as we go through and talk about corn nematode species. The longest one that we deal with, and it's interesting, the genus is called Longidorus, right? Long is in the name. And one of the most devastating that can cause damage in a matter of weeks is needle nematode. And needle nematode is about 8 times the length of SCN. So instead of a period at the end of the sentence, if you were able to hit the dash button, that's kind of about the length that we're talking about, but still not able to spot with the unaided eye. They're migratory ectoparasites. That's a fancy way of saying they don't crawl into the root, they crawl around on the root, and they jab the outside of that root with that needle-like mouth part going into the root and sucking juices out. So they're staying on the outside of the root rather than burrowing into the root. They have a long, hollow stylet. And these are a nematode species, partly because they're so long and they have to have big pore spaces to move within, that really like sand. So you'll see these in a lot of sandy locations, needle nematodes if you run into them. And rotation with this one can be a really useful way to manage needle nematode, but you got to make sure you keep grass under control, because if you have grass escapes, that's going to give some other root system for this thing to feed on. And remember, we talked about these are organisms that grew up feeding on grass prairie. So interesting here, just to make sure it doesn't sound all academic, you might be able, if you look very closely on the screen, see that little tiny line up towards the head. That's the stylet. That's the needle-like mouth part of Longidorus. We can go in just a little bit closer here in a bit and show you another example. Let's talk about sting nematodes. That's the next in size, about 3.5, 4 times the length of soybean cyst nematode. Again, a really devastating pest. Janelle's going to talk about this in just a little bit. She's going to talk about some of the numbers that we look for in soil samples that mark when a threshold has been hit. They are migratory ectoparasites as well, so they're crawling around outside the root, punching that needle-like mouth part into the root. They have a long stylet, and they also really, really like sand. Now, I'm going to circle something on the picture here just to show you some of that nematode anatomy at play, and if you look real close, you might be able to see that. If you lean in and look close at the screen, do you see that median bulb? Remember the little blue squeegee thing that they sent home with you from the hospital? And in the case of the nematode, something kind of like that helps create suction. There's that little median bulb on the screen right there. And if you look at the base, just on the top of that, you'll see the base of that basal bulb that creates those digestive enzymes that are squirted into the plant. You can kind of see those two structures actually in real life in this picture. After that, we have a nematode species that's about 3 times the length of SCN. It damages root tips. It has actually a long life cycle compared to a lot of other nematodes, and I'll come back to that in a second. It's also migratory, so it's moving around on the outside of the root and jabbing that stylet in, and it's called dagger nematode or xanthonema. It also is going to like sand. So the first 3 we've talked about here are sand lovers. They're a little bit more prone to reach those damaging levels in sandy fields although not completely restricted to sandy locations. They like those because they have those big pore spaces. Um, I mentioned this has a long life cycle. It's the better part of a year. I think some references will talk about a 9-month life cycle to go from egg to adult back to egg. Most of the nematode species that we talk about are like 24-day life cycles. So think about a month to go from egg to adult back to egg is about a month's time for many of the species. This is one that has a really long life cycle. It's going to prefer sand as well, and it doesn't like it if you disturb the environment. I want to zoom in here again. I want to point out there's that long stylet. If you look close here on the screen, or I'm going to pull actually up here a picture from the compendium of corn diseases that I have here in my library. And I want to point out, number one, you see the stylet, that's the long needle-like mouth part. Look at the base of that stylet, and you see those little knobs that we talked about. They're attached to muscles. And here's kind of an interesting feature. Right about in the middle, you can see there's some kind of little black line that kind of crosses that stylet. It's called a guide ring. The nematode stylet is so long it has to have a ring to make sure that needle keeps going through the mouth and it doesn't punch a hole in itself so that the nematode dies. It has a guide ring to hold that stylet in place so that it's going on the right path to go out through the opening of the mouth to feed on the target plant species. There's a nematode after that called Lance. Lance doesn't have to have sandy soils. So the first three really like sandy. Lance isn't really that way. Causes really deep damage to the root system, 2.5 times the length of SCN. And this is kind of interesting. This one both feeds outside and inside the root. And that means that sampling the root, at least at certain times of the year, as Janelle is going to talk about, sampling for nematodes at certain times of the year will sometimes require that you actually shave off some root material in your soil sample so that you can find these things. So there are some specific recommendations for different times of the year about needing to prune off a little bit of root material. Sometimes in the past, when we've done that, I actually will put that probe earlier in the season close to the plant, and I want to hear it cutting through root material because I want to see if I can find these things that are tucked away in the root system. Survives on various weed species, so rotation does nothing here to manage that nematode species. And again, a couple things that we talked about in our anatomy part here. Look up front. You see that little knob on the top? Those are the lips. Remember I said they create the suction that hold the nematode to the root system. You can see the stylet, that little vertical, and then you can see the little knobs down there at the bottom that are attached to the muscles that help move that stylet in and out of the body. And this one on the right happens to be a male because you see the bursa, that little flap, and you see those spicules poking out of it. There's a nematode species called spiral. Now we're getting closer to the size of soybean cyst nematode. Rotation's probably not really effective on this one. You can see why it's called spiral because it kind of curls up. It's going to like sandy soils, and if we get some really high populations, it's going to do a fair amount of damage. Then we have some non-common ones. There's several counties in southern Illinois, a few in southern Missouri, a few in southern Indiana, maybe a good swath of kind of eastern Arkansas and maybe western Mississippi that can see root knot nematodes that can feed on corn. Oftentimes we'll talk about root knot nematodes being more of a soybean pest, but those areas I described sometimes have a root knot nematode species called Meloidogyne incognita that can actually feed on corn and beans as well. It's not a real common nematode species, corn nematode species in our part of the world. It forms little galls. Just to keep pointing back at anatomy, there's the intestine on that thing. There's another not very common one called stubby root nematode. I'm just going to blow past that one for the sake of discussion. It's interesting, and it kind of violates some of those rules we talked about for plant parasitic nematodes because we said plant parasitic nematodes, most of them have a hypodermic-like needle mouth part. In this case, it doesn't have the hollowness to it, and its needle-like mouth part is kind of curved and it will slash the root. But again, not a very common species, not one for you to get really wrapped up about today. There's a nematode called stunt nematode, likes to feed on the outside of the root system, just getting pretty close to the size of SCN. There's one called ring nematode. It's not very common. One of the structures I didn't mention when we talked about anatomy, sometimes the nematode cuticle, its skin, will be reinforced by little rings that kind of make it a little bit more stout. It helps it stand up to punishment just a little bit better. And you can definitely see on this one, those little rings are what we call striations. Then there's lesion, and this one is really common, and that's the one we're going to wrap up with today. If you run a soil sample for corn nematodes, you're going to find lesion nematodes in those counts. It's really easy for us to run into those, and they can sometimes hit damaging levels about half the size of SCN. They migrate on the outside of the root. Again, punching that root system, feeding on it, cause some real distinct wounds that you can see there again from the compendium, these kind of dark brownish blackish wounds on the root system. And with that, I'm going to pause here and I'm going to close up with our must knows when it comes to corn nematode species, because we covered a ton of stuff, probably really hard to digest all of that. So I want to kind of wrap up with this. Here are corn nematode species must knows. You need to remember that there are some corn nematode species that are internal feeders, not just external feeders. They're actually going to bore into the root and feed on that corn root system inside. And because of that, as Janelle will talk about here in a second, we sometimes have to shave off some root material in the sample, not just soil, to be able to actually detect what species we're dealing with. There are some sand-loving nematodes that are a little bit more prone to cause issues on sandy soils. So needle, sting, dagger, and spiral. Those are your sand-loving nematodes. There's some nematodes that, frankly, you're not going to be able to manage with rotation. A lot of nematodes that we can, needle, dagger, some of those that we can use rotation to manage to pull back the population. But lance, lesion, spiral, and sting, and in some places root knot nematode, if it's that incognita version of root knot nematode, they feed on enough different things that a corn bean rotation isn't going to get them under control. It's not going to pull them back. And again, that's why this becomes just a little bit more complicated when we talk about corn nematodes, because we're talking about a bunch of different species that have little tweaks when it comes to management. And so genus and species ID is going to strongly influence the kind of management that we engage in. It's going to influence some of those cultural techniques that we engage in to pull the population back. So again, walk out of here one more time. Nematodes are microscopic tubes of water with organs floating within. Plant parasitic ones are feeding on the root system with a needle-like mouth part, partially digesting material outside the body, sucking it in. We have female and male nematodes. Sometimes it will be exclusively female. We have a life cycle that goes from egg to juvenile stage to adult to egg, oftentimes about a month for it to do that. We have different types of nematicides. We have a whole bunch of different types of species that we deal with when we talk about corn nematodes. And you need to remember that some of those like to feed inside. And you need to remember that species and genus ID is kind of important. That's why we do the soil sampling, because it helps us better hone in on the management techniques that we need. And with that, I'm going to push escape here. I'm going to stop sharing my screen, and I'm going to hand it over to Janelle.
Hi, everyone. Give me just a second and I'll share my screen. Hopefully that's sharing for you all in PowerPoint format. So today we're going to touch base a little bit around the nematode sampling that Matt had mentioned, and then go into a few of the different management strategies. Like he mentioned, just the fact that there's so many different species with corn, as opposed to when we think of management strategies for soybean cyst nematode, you've kind of got to tailor based off of your results from your soil sampling. I do want to, he talked briefly about some of the symptoms. I do want to touch base on the, kind of the damage that we see from not only just nematodes, but some of the diseases in general when it comes to corn. So this is a graphic from the Crop Protection Network that they put out from their 2023 review. This is looking at the 29 US states and Ontario in Canada, accounting for the bushels lost due to disease. And we can see that in 2023, these folks that feed into this survey, they determined that nematodes, corn nematodes, was the #1 hitter when it came to loss of bushels. So this whole schematic, all of these different diseases and issues account for 465 million bushels of loss due to disease. And the nematode section accounts for right around 56 million bushels. So I think we hear a lot about tar spot and fusarium and some of these other things, maybe specific to your area. But one thing that I think that this graphic points out is that nematodes, even though we're maybe not talking about them a lot, that's an issue that we're seeing. And it's really kind of depending on the time of year, or depending on the type of year that we had, we see nematodes kind of rise to the top. So I just wanted to share kind of the impact of nematodes on corn in the US and Canada. When it comes to sampling for nematodes, Matt kind of indicated that there, might be some time to get roots, there might be some time to get soil. So when we look at this schematic down here of the corn growth and development, if you're looking at corn VE to V6, that's when it's also helpful to get soil as well as root samples. So you can dig up anywhere from 4 to 6 roots, and we'll talk about that here in just a few steps, but to get root samples as well. When you're V6 to R3, it's best to collect soil samples. I'm sure you can send in the root samples and they may be able to help diagnose things based off that. But really, the diagnosis is going to come from those soil samples. When you're looking at taking soil samples, how might you do it? It's best really to have a probe. I know that's not always the case, but if you do just have a spade, or if you're lucky enough to have a probe, make sure that you're going at an angle through the corn's root zone. That's where Matt talked about, hey, let's try and get some of those root hairs, because some of those are inside the root feeding and causing issues. You're going to want to go 8 to 12 inches deep into that root zone, and you're going to want to collect 15 to 20 cores. That way you've got, and do it from a couple different areas in the field based off of, maybe symptomology, or you've seen a yield drag in a certain area. Make sure that you're from a couple different areas in the field. For those plant samples, if you're looking at VE to V6, dig up some plants, knock off the dirt, and make sure that we're putting those in a separate bag from the soil when you're getting ready to ship those out. So really, based off of your growth stage in corn, that's going to tell you, should I get just a soil sample, or should I also send a plant sample as well? I know that we've got in here, okay, VE to V6, V6 to R3, but I'm here to tell you, you can also sample in the fall after things are off, okay? Because that still gives you some sort of baseline. Obviously, it's best when you've got corn growing in the field, but you can also sample in the fall if that's when a grower has time to be sampling. You're going to then combine those cores into a sealed plastic bag, once again, separate from the plant sample, and then label them. One thing, probably the key thing to note, is that if you are not going to be shipping these out right away, you need to store these samples in a refrigerator. So we don't want to throw them, on the floorboard of the truck and say, okay, I'll get to that in a few days. I'll ship those in a few days, because then your sample is going to start to degrade. They need to be in the refrigerator and then taken out as soon as you can ship them. When it comes to shipping to a lab, there's a number of different facilities that test for nematodes. We're lucky in the fact that most of our universities still offer this either through their nematode lab or through a diagnostic clinic. One thing to note here is that we're looking for a plant parasitic nematode test, not just a soybean cyst nematode test. So if you happen to check the box for just SCN, they're only going to look for that species. Remember all the slides that Matt just shared around the numerous species, dagger, lesion, stunt. We need to get a plant parasitic nematode test so that we can get the results for all of those. I went through a couple of different universities and I just wanted to see about how much this would cost. These are for in-state sampling. So for instance, if I, who I'm a resident of Indiana, were to send a sample to Michigan, I think generally out of state is going to be $10 to $15 more than what is listed here. But obviously you can touch base with your local university. Most of the time when you go onto their sites, they're going to have a review of how to sample, how to send in the sample, how to pay for the sample. That way you can make sure that your results get back to you as soon as possible. There's also commercial labs as well. So you can, I've worked with Midwest Labs in the past. SGS has a nematode sampling protocol as well. And I know that there's other labs out there that are also doing sampling. I just didn't have the time or bandwidth to Google all of them. But we really just wanted to get an idea around how much this would cost if you guys were interested, or if you had folks that were interested in sending in samples. So you've done your sampling, or either you've seen symptoms or you just have a field that you've had some yield drag in and you're wondering, hey, what's going on with this field? And so you've pulled the soil samples, those 15 to 20 cores, you've bagged them up, you put them in the fridge, overnight, and you've shipped them off. You've done the best that you can, right? And you get your results back from that lab. There's a lot of information that goes into these results. I'm going to point out right up by the yellow star, this is for a plant parasitic analysis. So it's not just the SCN, make sure we're going for the whole gamut analysis. Sometimes through some of these universities, it'll be listed as like a turf nematode analysis, plant parasitic analysis. So that would be the same, that would be the one that you would be after when it comes to corn as well. When we look through this list, you're going to see a number of these that are, familiar for what Matt just told us about: lesion, stunt, spiral, dagger. And I want to go through at least 3 of these that I've got listed up here and look at the damage thresholds and how this sample that I sent in the fall of 2019, how it stacked up to those thresholds. Okay. So understanding the thresholds will help you understand some of the management you need to take into account, or, maybe, hey, I need to make sure that I'm working this field first and then, then go to this field that's got the higher count numbers as a cultural practice, as a management practice. That way, that last field is my field that's got the most the most nematodes, and so I don't want to drag stuff around. And so that's why I think some of these getting soil samples is a good idea, because it gives you an idea how you can best manage them. So if we look here at spiral, it says that we found 212. Well, if we follow the line down to spiral, that's going to be in the green because our threshold is 500 to 1,000 per 100 cc of soil. And so we're good there. But when we drop down to dagger, we've got 106 dagger nematodes in that sample. When we go to the thresholds list, the damage threshold for dagger is 30 to 40. So the threshold is 30 to 40, and we've got 106. So we know that dagger nematode is an issue for us. Drop down to needle. Needle nematode, we've got less than 26 is kind of how it's their reporting limit. But when we look over at the threshold for needle nematode, which is the biggest, the longest nematode, right? The threshold is very low. it's only 1 per 100 cc of soil. And so we know that needle is an issue for us, as well as dagger. Spiral is, you're at 212. It's something, obviously you've got some spiral out there, but it's not of concern from this sample. I also wanted to take a look at, this is a study that Syngenta did back in 2008, and the graphic is not all that great. And I know that there's been updated surveys since, but I really like the map on this one because it helps indicate that, these corn nematodes are not just clustered to one region. And in fact, if you look at Iowa and Illinois, where they were able to test, most of them are at low, which would be yellow, moderate, or high, which would be red. So most of them in that state where they were able to get to each county and test, they were able to find moderate to high densities of nematodes, corn nematodes specifically. And this isn't necessarily a scare tactic by any means, but this just goes along with the fact that, these corn nematodes, the 12, 15 species that really impact corn, they're grass feeders. And so in the fact that most of our marketing area was prairie at one point, that means that now that's just converted over to a corn nematode. And that goes into a little bit of the management as well. So touch base on some of the management. The cultural practices, and Matt was great, and he went through for each of the nematodes. He had a little bit of information around cultural practices. Unfortunately, there's no silver bullet. I can't give you a product that really blows everything out of the water. I can't give you a certain practice that blows everything out of the water and you don't have to worry about nematodes. He mentioned, some of those nematodes like sand, some of those nematodes the tillage helps with, some of those the tillage doesn't do anything for. A lot of these are just looking to bolster your crop so that a healthier crop, a healthier plant can help fight off the nematode a little bit better than, say, a plant that's already starving for fertilizer or it's already starving for water. So planting date is, the idea that maybe you plant a little bit later. That means that crop can get up and out of the ground, really put on some roots because of the warmer weather and kind of outpace the nematode feeding. I'm not sure how much that actually helps, other than the fact that there are some that, maybe you can kind of outpace. Tillage, some of them, some of the nematode species can be interrupted due to tillage. So really you're looking at reducing the compaction. Crop rotation, some species are also a pest on soybeans or also a pest on cotton. And so occasionally crop rotation isn't a help, but in some species it would be. Weed management, Matt went into the fact that a lot of these are grass nematodes, right? So we can help keep our fields clean at the beginning and end of the season, because one of the things is that the life cycle on some of these nematodes is 24 to 30 days. So if you've already got some weeds out there, especially grass species out in the field ahead of that crop being planted, you've already allowed that population to start reproduction. And so you may, especially in a year where there's some delayed planting, unfortunately, you may be one generation ahead of what you could have been had the weeds been properly managed. Irrigation and fertilizer, that just goes back to making sure that we're feeding that crop just to really bolster it so that it can outpace, and a healthier plant can help kind of bolster against the nematode invasion. And then sanitation, that one in itself really just goes back to, hey, I know I've got this nematode field, let's till that last, let's plant that last, or kind of spraying down equipment in between fields. When I was in grad school, we used to, the booties that you can put on your shoes if you go into a business, or if you've got like an AC repairman that comes to your house, generally they'll put those little booties on. Well, we used to if we were going to a hot nematode field, we would then wear booties in to make sure that we weren't getting dirt on our boots, and then we would wash our boots off. And then when we went into a different field, we would wear a clean set of booties so that we're making sure that we weren't moving nematodes field to field, especially because generally when we were called out there, the situation was probably pretty dire. It kind of felt like you were walking on a bed of nematodes, right? In some of those high count areas. Host resistance. When we think of soybean cyst nematode, the last few years, we've heard quite a bit about host resistance or just resistance backgrounds like PI 88788 and Peking. That's not really something that comes into play for corn nematodes. One, because there's a lot of different species that all of the companies would have to be screening each and every hybrid against, and that just takes time. And really nobody's set up to do that. On the soybean cyst nematode, that they've kind of got that down to a fast science these days. And really on the corn side of things, it's more around tolerance than resistance. One way that you can kind of utilize host resistance is utilizing resistant cotton or soybean varieties when you are rotating away from corn. So really just trying to kind of beat down those nematode populations when you're on an off crop. There are a number of different nematicides out there, and there's a number of different applications when it comes to nematicides. Nematicides are going to protect your root system for 30 to 45 days. You may see that go, longer. You may see that only be 15 days. Really, that comes down to your soil type, the weather, kind of what that product is going to be put through. I would love to be the one, it's my job at Beck's to oversee our seed treatment package. I would love to tell you that I could give you a seed treatment that is going to protect your corn crop from the day of planting through the day of harvest, but that's just unfortunately not the case. So some of the options that we do have though are in furrow or starter additives, and I'll talk about a few that we've seen in PFR. Talk a little bit about seed applied nematicides, both biological and hard chemistry. We'll talk specifically about what Beck's offers with Nemasect. And then there are also some planter box options as well that are biological in nature. And so those are a lot of times are the ones that wouldn't last on seed. So their on seed life would be short. Once they are introduced to water, then that it's a living subject. So it wouldn't last, maybe more than 24 to 48 hours after it's been on the seed. So that's in that instance, those products are best used as a planter box option. In 2023, PFR had a corn starter additive study, and it was a number of different locations. They had a couple different products in here, but I'm pointing out two that have that are labeled as nematicides. So the Zionar product, which is from FMC, it's labeled as a biofungicide and a bionematicide. Okay, it's two different bacillus strains that are going after the nematodes. Averland FC is a hard chemistry nematicide, and its AI is abamectin. I do want to point out that both of these products do have seed applied labels. So Zionar, it has a label for seed applied, but it is under a Votogen brand. And then Averland FC, the abamectin, would be a Victa as a seed applied product. But anyways, so if you know PFR is our practical farm research program here at Beck's, and we're really just looking at trying to find products that help the farmer's bottom line, really. So if we look at this study for multi-location in 2023, we can see the control is just starter. Then you've got the starter plus a product called Xylem Plus, which actually won in 2023. But the two that I'm interested in would be the Zionar and then the Averland Plus or the Averland FC in addition to the starter. And we can see that both of those products are providing a return on investment around 3.5 bushel over the control. And these are at, like I said, multi-location. So we're going to have them under varying corn nematode density pressures. And so we can see that these two products in itself as a starter additive both pay when it comes to helping with corn nematodes. In 2024, the corn starter additive study through PFR, it also had Zynar, as well as a couple other products that were not necessarily nematicides. But I did want to point out the Zynar again, because that was our third year of testing in PFR. And so for PFR, if a product has 3 years with a positive return on investment, we then deem it to be PFR proven. So in 2022, we tested this product at multiple locations and we saw the return on investment of $4.35. 2023, we saw that return on investment jump to $9.46. Unfortunately, in 2024, that dropped from down to the $6.57. So the average is $2.41 advantage for Zynar. And this isn't necessarily a plug for this product by any means, but I'm just letting you all know that there are some options beyond a seed treatment option as well. Like I said, with the starter additive and or the planter box option. For Beck's, we have our Escalate seed treatment package that goes on all of our corn hybrids, and it's made up of a plethora of different products. So that could be products, fungicides, insecticides. In this case, we've got the nematicide Nemasect. So that's what we're going to kind of focus in on today and give a little bit deeper of a dive into Nemasect as a whole, and then how it works against certain nematode species. So Nemasect is derived from a bacterium called Burkholderia, and it is classified as a biopesticide. What's interesting about Nemasect is that it is not living. So it is a product of fermentation. So it's a biological product that they then ferment, and it's that fermented media that is what works against the nematodes. And so it handles more like a traditional chemistry, in the case that, if you have a live bacillus and it's a nematicide that you're putting on, it really has to have decent conditions for it to thrive and for it to protect your roots. So that's one of the things that the Beck saw in Nemasect when they first launched this way back in 2018, I think it was, is that it handles like a traditional chemistry. So we don't have to worry about Nemasect, the life on seed. So a lot of times I mentioned some products really just have to be a planter box additive because they won't live on the seed beyond 48 hours. That's not the case with Nemasect. It handles like a traditional chemistry. For the modes of action, so most of the time it's going to be working through enzymes and toxins. So killing those nematodes within 4 to 48 hours of coming into contact with them. A few that are on the label would be sting, needle, lesion, stubby root, dagger, and root knot nematode. That would be corn plant parasitic nematodes for corn. We also have a long-standing study where we're comparing Nemasect to a standard fungicide and insecticide treatment. So in this case, we've got the standard F&I treatment versus the standard F&I treatment plus Nemasect. So what we've seen over the last 9 years is a yield advantage of over 9 bushels per acre. And this is kind of a broad range of regions. You can see down here in the footnotes, we're looking at Nebraska, Minnesota, Missouri, Iowa, Illinois, Indiana, Ohio, and Alabama at 240 different locations. So what I like to do with the piano graph is I like to, take the middle. So I'm cutting off the bottom and I'm cutting off the top, and I hone in on that middle. And when you hone in on that middle, you're looking right around 4.5 bushels per acre advantage to having Nemasect on corn. Diving into some of the specific nematode data for corn, we worked with Mississippi State University back in 2017 to look at some greenhouse and growth chamber studies and as for how Nemasect really impacts some of these specific nematodes. So what we can see here, this is the number of nematodes per gram of root. So we've got our F&I check on the blue bar, okay, right around 1,500 nematodes per gram of root. And then you can see the F&I check plus the Nemasect product, and that dropped it all the way down to right under 500 nematodes per gram of root. So that's what we like to see. And we like to see in the fact that we know that it's got efficacy against this specific nematode. These were, like I mentioned, they were lab or growth chamber studies. They were inoculated with root knot nematodes, and then they were harvested at 60 days after planting. In 2016, and the reason for 2016, 2017 is because Beck's chose to move to Nemasect in 2018. So a lot of the initial work that was done was, 2014 to 2017, looking at the specific nematodes that Nemasect would impact. So this one happens to be from 2016. The University of Nebraska did some work with lesion and sting nematode. So here we're showing bushels per acre, so the yield. So no nematicide is the blue bar at 181 bushels. Then we've got a competitive check at 197 bushels. We can see Nemasect in the mix at 208, and then another competitive product at 188 bushels. So really what we like to see here is the fact that obviously we're helping out compared to having absolutely no nematicide, but we also like to make sure that we're doing well in comparison to other nematicides as well. One thing that's interesting about Escalate is we kind of have that option to add different products in. So we're always evaluating different products and making sure that what we're currently offering is either at or above what some of those other offerings would bring. Here's 2017 data from the University of Nebraska. This was just a look at lesion nematodes. So in this study, they just happened to, the soil sample really didn't show too much of the sting this time. So we've just looked at the lesion. Same setup, this is bushels per acre. We can see having no nematicide is 237 bushels compared to having Nemasect added in at 245. And then the two competitive products here down at 230. You might ask, why would those two competitive products be a little bit lower? I would, without kind of divulging everything, those are two biological products. So if there's a lack of moisture in the soil, a lot of times those biological products don't necessarily work as well. And so that's where it's nice that Nemasect works more like a hard chemistry, being that it's heat killed. And so that we still saw that benefit out of having the nematicide for Nemasect on compared to a biological that kind of struggles in some weather conditions. That is what I'm going to end with for today. I know that that was a lot of data, kind of technical review. Matt is going to hop back on and share some of the QR code with the CCA points, and then we will answer some questions.
Okay. I think everybody, can you see the QR code on the screen there?
It might be best if you put it in presentation mode, Matt.
Okay, will do. I'll do from this slide. Right there. How does that work, Janelle?
There it goes. Yep. Okay, now that should be much easier for people to see.
So Steve, do we have any follow-up questions or anything like that from the group?
Oh yeah, fantastic presentation. Thank you guys very much. And Kyle, I think I can catch what's in the chat. If I miss something, let me know. But You guys had talked about, a lot of the different management practices, Nemasec, those options, and you kind of hinted at some of this tillage. And sometimes tillage can help, sometimes it's not. Are nematodes more prevalent in no-till versus conventional till? Does it matter? And then to follow up on that, if I am in no-till, is there any advantage or disadvantage to having cover crops, with most of them being a grass species, to nematode populations?
Well, that's an interesting one, and Janelle, feel free to jump in here as well. As far as tillage goes, I think so. One of the things to keep in mind, and we really didn't talk about this, is that nematodes on their own aren't going to travel very far. I think some of the numbers that I've looked at, I mean, maybe you're lucky if they're moving a couple feet during the growing season, and a lot of times less than that. And so a lack of tillage probably means that we're doing things that keep the nematode species from being moved around as readily. You may remember Janelle mentioned a little earlier in her presentation that if you get a real high count, you start triaging that field as kind of being the last one that you go into, so you don't spread it around. And so I suppose from that standpoint, that a lack of tillage might actually help us not move the pest around as readily, might help us not have it as widely distributed through a field where we kind of drag it out as we're going through with tillage equipment. But I think the qualifier there would be, once again, that a lot of that is going to depend on weed control. If you let the weeds get out of hand, I'm not sure that all of the benefits I just talked about are actually going to show up. Janelle, do you have any thoughts to share on the topic?
Yeah, I would think that the weeds, especially around cover crops, that can have an impact on nematode species. I worked more on soybean cyst nematode and cover crops than I did just kind of a broad look at nematodes and cover crops. But one thing that we've seen, right, is that especially like cereal rye, occasionally that can increase your some of your nematode counts. Um, so that's something to take into account, maybe making sure that we're getting those killed off in a timely fashion so that we're getting the benefit from those cover crops. I'm not saying that cover crops are bad by any means, but make sure that we're getting the benefit from those cover crops, but then also not having additional plants out there before that next corn crop is introduced, so that we're not kind of getting an early start on those nematode generations.
And Janelle, I guess I should throw in there too, it depends on the cover crop as well, kind of like you mentioned. If it's something like rye grass, you can see where that type of root system would be a lot more attractive. There may be a bit of a hint of people wondering, like sometimes in the past we've talked about certain mustard crops tilled under will actually help decrease nematode populations. I know that's not necessarily what we're talking about in a no-till situation, but again, there's a lot of variability depending upon the species that we're talking about, not just the species of nematode, but the species of the cover crop.
Thank you. Another question came in. You had mentioned a few times about rotation, just kind of in passing, that rotating crops can help pull down populations of some species, but then others it didn't affect.
Yeah.
So in a situation where I've got corn nematodes and I go into a 3-year bean rotation, which some of the Beck's marketing area can be that way, long-term beans, would we expect a decrease in those nematodes if we had good weed control? Or is there other things, or their longevity in the soil longer than that?
Yeah. So again, great question, Steve. So again, some of that depends upon the species that we're talking about, and that's one of the reasons that a soil analysis is important. It's not just important to know whether you have threshold levels or not, but that really does guide whether rotation will prove an effective strategy. So like lance, lesion, sting, spiral, those have a wide enough host range that rotation's not going to matter with those ones. It's not going to matter in those cases. They're going to have enough other things that they can feed on. The rotation's not going to pull that population back. And I'm not sure if I remember the second part of your question there, Steve.
Well, just if I was in a long-term soybean crop, would I expect a drop in populations of corn nematodes?
Yeah. So again, it kind of depends on the species. For instance, that if we're talking about this obscure one called Meloidogyne, root knot nematode, those things, rotation knocks that population down substantially. Like I think Dr. Eisenbeck has said something like 90%. We don't deal with this type of corn nematode, but corn cyst nematode, which isn't a little blip of an area on the East Coast, that thing, it's going to survive actually extended periods of time, season to season. So it again goes back to this topic, I think, of that corn nematodes is a little bit more complicated product, a little bit more complicated topic than maybe something like soybean cyst nematode is, because we're lumping a bunch of different critters under one category. And how long they persist in the soil really depends upon the species that you run into. Rotation should have a benefit on many of those, pulling it back, but it really is going to depend upon what the population that you're dealing with, what the big players are in that population that you screen.
Very good. Well, that looks like, from what I can tell anyway, we're caught up on questions, and I know we're at our hour and a half. So I really appreciate Matt and Janelle taking their time, sharing their expertise. It was a fantastic presentation. Thank you, guys. And if you've got other questions or questions come up after this that, boy, I wish I would have asked that, reach out to your local Beck's representative, reach out to your dealer, your seed advisor, your agronomist. Matt and Janelle are also excellent at answering questions. So we'd love to help you out any way we can, and we really appreciate everybody joining the presentation today. Any other last thoughts, Matt or Janelle?
I don't think so. Thanks everyone for joining.
Oh, wait, I did have a question just popped in.
Oh.
If you're still there.
Yep, still here.
Yeah.
If we put biologicals and insecticides, won't the insecticides kill the biologicals?
Yeah, that's a good question. Some things when you are putting especially hard chemistries on the seed, really you have to make sure that that you're whatever biological you're putting on the seed has to be more of like a spore in spore form as opposed to something that's already germinated. So a lot of the times that's some of the evaluation that I'm doing behind the scenes to make sure that if we're putting on a biological, we want to make sure that none of the other products that we've already put on, like mefenoxam or thiabendazole, that they're not going to be impacting those biologicals. It's kind of a dance to make sure that we can keep some of those hard chemistries on because they do a lot of the work for us when it comes to pythium and fusarium, but also we want to make sure that we are have the ability to use some of these biologicals because the biological thing is really kind of exploding over the last few years. And there's some really interesting ones out there that we want to make sure that we can use and utilize on the seed. But you're correct. Sometimes there is some negativity between a biological and an insecticide or a fungicide, but most of the time that work is being done in advance of what we would put out at Beck's in our Escalate package.
So ultimately, Janelle, what you're saying is from our standpoint on the seed treatment part of Escalate, you've done all that research, you know they work together. But if I want to apply a biological, maybe that's in furrow, that's a seed box treatment, I need to be asking questions to my supplier of that product of what that effect of the insecticides might be to make sure I'm getting the benefit.
Yes, and most of the time it's very common practice for those folks to have already tested against all of the common seed applied insecticides and fungicides. Because you know, a good majority of the corn industry is sending out their corn with either you know, poncho or clothianidin or thimethoxamone. So they want to make sure that their product is compatible before they do sell it to other folks. But yes, for sure, check with your supplier.
Okay, fantastic. All right, I think that got us caught up on the questions. So again, thank you everyone for joining, and we really appreciate the interaction, and thank you again to Matt and Janelle. Everyone have a great day.